Thank you so much, folks, for joining us for our final formal presentation of the day. Dr. Stephan van Vliet has dropped his bio as well of Utah State University, has dropped his bio as well in the session information. So we hope that you do check into his background and past work. And I'll turn it over to Stephan to begin his presentation. Thank you so much for joining us today.
Yeah, thank you all for joining us today, and thank you so much for the invite to present my work. So I have a particular interest in systems research and understanding linkages between plant, animal, and human health, and even soil health. And we've been using a technique called metabolomics, where we really take a holistic approach and we profile several hundreds of nutrients across these biological systems to see if there is a relationship between the health of plants, animals, and humans. And this is what I'll talk about with you today, specifically as it comes to grazing systems. These are some of the disclosures, current research partnerships with USDA, NEPC, ARS, the Turner Institute of Eco-Agriculture, the Dixon Water Foundation, and NCBA. You can also see my conflict of interest for the food policy below. So we're broken up into three major parts. Part one, first we look at sort of a bigger picture discussion, understanding relationships among plant, animal, and human health. And then we will go into the metabolomics data. We can tell something about animal health from that, as well as the nutritional qualities of meat. And we go far beyond just omega-3 fatty acids. So oftentimes we hear this relationship that healthy soils equals healthy plants, equals healthy humans, or healthy animals, and equals healthy humans. While that intuitively makes sense, and there are certainly pockets in the literature in their respective fields that would suggest that, there has been a lack of systems research that tries to tie it all together. And I'm very excited about this. The way we're heading in the field is that we're seeing soil scientists and livestock scientists, agronomists, economists, and nutrition scientists, such as myself, all work together because we have to share the same goal ultimately, and that is how can we nourish a growing population. So Dr. Jason Roundtree already laid it out very nicely this morning for all of you that were on his talk, but to just give you a bigger picture is that how difficult it is right now also for farmers and consumers, and the grand challenges. There's large concerns about the effects of food production on planetary health. And we also have now this issue and where we started after the Green Revolution, and it made a lot of sense, is to use simplified high input systems to produce food as efficiently as possible. Now, as humans, we always like to simplify things. I think that's the main way for us to understand it. But in contrast to that, and we talked about this in a recent paper that we published about closing nutrient cycles for animal production and how to move forward to produce foods in healthy, sustainable, and ethical ways in the next 40 to 50 years. But one of the things was, and that always struck me, is that nature has introduced great biodiversity, but us as humans are adamant about simplifying these systems to single species of plants or animals. And as a result, they require considerable external inputs. Now, as I said, that perhaps in the past made a lot of sense, but right now we also need to start thinking about how can we do this in another 50 or 100 or 200 years from now, because simplified systems are often productive in the short run, but they make it difficult for farming systems to recycle nutrients and be sustainable in the long run. And these are often discussions that we have, and I'm on a panel for the FAO and the Scientific Advisory Committee, where we look at livestock in healthy and sustainable diets. And really, we're trying to take a holistic approach there too. It's that we're often focused on things like greenhouse gas emissions, but there's other things that we need to think about too. And the 5 principles of sustainable food and agriculture are: 1, improving efficiency and the use of resources. Obviously, we need to conserve, protect, and enhance our natural ecosystems, but we must also consider the farmer and rural livelihoods and social well-being, of course, the resilience of people and communities, and also promoting good governance about natural and human systems. So these are some of the environmental challenges we have. And as a consumer nowadays, it is not easy either, because every week we get slapped with a headline with concerns about red meat on human health. And it seems that most scientists rarely agree upon things. And there's a huge controversy at the moment whether we should even consume red meat in our diet, as is suggested by some. So we get slapped with headlines: red meat eaters have a higher risk of dying from several diseases. Then there are also other studies. There was a group of researchers in the Annals of Internal Medicine that suggested that, hey, maybe this data that we've been studying regarding red meat, maybe that data is not as strong as we thought it was. It is largely epidemiological associations with relatively small relative risk factors. So another point that's often forgotten is that most of the time we're studying red meat as part of a standard American diet, whereas various other studies, also epidemiological studies, suggest that when consuming red meat, either grass-fed or grain-fed, which is what most of the studies have done, grain-fed, is compatible with good health when consumed as part of a high-quality diet, also rich in fruits, vegetables, fiber, and low in ultra-processed food. So oftentimes this is also studied in the context of a standard American diet. And so there's always conflicting headlines in the news, which I can only imagine how hard it must be for a consumer to sift through that. But important to note, oftentimes these are epidemiological associations. People are asked what they eat. So for instance, one question might be, how often do you consume red meat over the last 3 months? People are followed for a long period of time, 10, 20 years, and then we see how they develop, if they develop heart disease or cancer or all-cause mortality, which is then linked back to the food frequency questionnaire, often asked several years back. As you can imagine, lifestyle factors play a role here too. But just to paint a broader picture, while nutrition and scientists and climate scientists argue on whether we should move forward with livestock production and the role of livestock and red meat in our diet, we also must consider farmers and communities, obviously. And this is really where my interest comes in, is there is an increased interest of consumers and farmers in, especially small-scale producers, in pasture-based systems that aim to improve soil health, plant diversity, animal welfare, climate resilience, and reduce farmer input costs and improve the quality of life as farmers. And oftentimes this is referred to as regenerative agriculture. And as we can see, there's also consumer interest, especially in pastured meat and milk. So these are the projects that we're working on, and I'll jump into it right now. Is that the data that I'll present today is from a USDA SARE-funded project, where we are starting with soil. We move to plants, animals, and humans. So we're linking production to human health, and we call it the project From Farm to Table to Us. Essentially getting after the very simple question: do more sustainable practices also improve health along this continuum? By improving one, do we improve the other? So our study involves soil collections, and here on the left you can see a conventional cornfield and a biodiverse pasture. So the corn would often go into commodity markets, so this was our model for sort of the conventional grain-fed system, and here's biodiverse pastures. So we sampled those, see what the impact was on soil health. And then we also collect feed and forage samples. On the left, you can see a total mixed ration sample. On the right is a pasture sample. Those are clearly different, but we profiled the nutrients in the soil and the biological activity. We profiled the nutrients in the forage and the feed. And then finally, we also collect meat samples from all these farmers. And we do deep metabolic profiling to study sort of life along this continuum and look at nutrient density and biological activity. So, which brings me to part two. What is the impact of grazing practices on the nutritional qualities of meat and animal health? Some of the other speakers talk more about it from the standpoint of soil health and soil carbon sequestration. Well, I will focus on the nutritional qualities and present this portion of our work. So the study that we've been doing is we do on-farm research, and this was performed last year during the grazing season. We go to farms, we collect soil, we collect plants, we collect meat. In this case, the purpose of this was to determine difference in metabolomes, which are nutrient profiles and animal health markers of grass-fed versus grain-fed beef. And the grain-finished beef was obtained from a Midwest feedlot, and the animals were finished on a corn-based double mix ration, as is common. And then the grass-finished beef was obtained from Alder Spring Ranch in Idaho. And the farmer herds his cattle on biodiverse pastures during the grazing season, consuming probably up to 100 to 200 different plants on the pasture. So it's very phytochemically rich. Now, we took those samples. We got 18 ground beef samples from the pasture-based system and 18 ground beef samples from the grain-fed system. We cooked those in our metabolic kitchen. We sampled those and then we analyzed those. So this is the study workflow that we used. So we used a technique that is called metabolomics, and I'll talk to you a little bit about what metabolomics is in the next slide. But basically, we take these samples, we take them into the lab, we process these samples in our wet lab facility, and then we run them on what is called a mass spec. So what a mass spec does, it is used for all kinds of applications, also, for instance, even Olympic athlete drug testing. But basically, in biological samples, such as meat, you would scan the sample for an hour, and every nutrient or metabolite has a unique molecular mass. And at various times throughout that scan that takes an hour, you see a peak occurring that corresponds to a very unique nutrient or metabolite. Now, we analyze these metabolites, and we typically identify 500 to 600 metabolites. And obviously, when you have such a large data set, we do a bioactivities and pathway analysis. We group these in polyphenols, amino acids, and then we interpret the data using various databases to come up with an indicator of animal health and nutrient density. Now, this is different from sort of the traditional methods that have been used, because when looking at grazing systems, we often looked at omega-3 fatty acids. And it has been well established that omega-3 fatty acids are enriched in pasture-raised beef compared to grain-fed beef. Oftentimes, it's also thought that those are potentially the only differences. There is indeed not a lot of difference in terms of protein and in terms of maybe iron and zinc and several of these other minerals. But it's only a handful of nutrients that appear on food nutrition labels, and we're often very much focused on that. But the food matrix contains hundreds and hundreds of bioactive compounds that can potentially impact human health. Oftentimes we hear, for instance, saturated fat is bad for you, right? But saturated fat is a very broad category with various saturated fatty acids. So what we are doing in my research group is we are connecting the presence of these unique compounds in foods and the dietary patterns with the human metabolome, studying how the biochemical complexity of these foods and impact human health and or disease-related gene expression, which is called nutrigenomics. And by overlaying the food metabolome and looking at all these unique, potentially health-promoting compounds in foods with the human metabolome, we can get an insight into how our food choices impact our health. And the meat metabolome is then also compared to soil health metrics and also the forage metabolome to see this transfer of nutrients along this soil, plant, animal, human health continuum. And it allows us to do so in appreciation of the whole food matrix, because classically we looked at single nutrients and human health, but we are really looking at more of an holistic approach, like, okay, what are all the nutrients that are in foods? How are they impacted by production systems? And how do they impact our health? And what is important to note is that we often hear this comment, you are what you eat. Well, there's half truth to that, because about 50% what circulates in our blood is directly related to the foods that we eat. So when we look at the data, this is the statistical summary, is that we compared 18 grass-fed beef samples with 18 grain-fed beef samples from a feedlot and a biodiverse pasture. we identified nearly 600 biochemicals. Of those, and this was even surprising to us, 65% of them were different. So these differences go far beyond just omega-3 fatty acids. We found that 286 of them were higher in grass fat, 91 of them were higher in grain fat. We also did a lipid profiling. We looked at almost 1,000 different lipids and fatty acids. Again, far beyond omega-3 fatty acids, and we found that 53% are different. 307 higher in grass and 223 higher in grain. I do want to highlight that higher doesn't always mean better. It depends on the biochemical. For instance, we know that certain fatty acids associated with metabolic disease and others do not. So higher doesn't always mean better per se. It just means that they are very much different. So if we look at the data explanation here, and this is the data that I'll present, just for ease sake, green means higher in grass fat, red means higher in grain fat. And the data is expressed as grass divided by grain. So in this case, alanine, which is an amino acid, will be 1.8 times higher in the grass fat beef compared to the grain fat beef. All right, so when we jump into the data, so I told you we come up with these 500 metabolites, right? So now we need to make sense of the data. And this is sort of a high level overview. What if we put these 500 metabolites in a software program and we let the program figure out which samples cluster together? What we can see here on the left is that the grass fat beef samples cluster together. On the right, the grain fat beef samples cluster together. What we can see from this is that grass fat beef is in grain fat beef, and grain fat beef is in grass fat beef. There are substantial differences. We will now look at what these differences are and drill deeper into that. What I do want to highlight is that when people ask me, Stephan, how can you be so confident about these findings? Well, if we use machine learning and you have a random forest confusion matrix, we basically put these samples in the software and we ask, or we let the program figure out which samples cluster together. So it predicted with 100% predictive accuracy which samples were in the grass fat group and which samples were in the grain fat group. So we can be quite confident that these differences are real and not due to chance. So the top 30 ranking biochemicals that separate grass and grain fat beef are vitamin metabolites, phytochemical metabolites, and lipid metabolites. And we will talk about these individual compounds now. And we also, as you can see up here, X25950, it means that we have no idea what it is yet. So we have some work to do in the future. But for ethylphenol sulfate, benzoyl carnitine, those are phytochemical metabolites. So from polyphenols, phenolic compounds that have potential anti-inflammatory and antioxidant effects, both for the animal and potentially to us too. So if we start with the animal health markers, not unlike human muscle, the muscle of animals, they provide many clues regarding the metabolic health of the animal. So I'm originally trained as a muscle physiologist, specifically in humans. And what we do in my lab and still do is we would take muscle biopsy samples from people. We take a small piece of their muscle and we do similar analysis as we've been doing here, metabolomics analysis. Muscle is the largest organ in our body, and it gives us a very good indicator of the health of our overall health overall. Cows are mammals too, so we can study cows and the muscle of cows, meat, and also say something about their metabolic health. And what is interesting, if we start with glucose or energy metabolism, if you've ever been to the doctor and your doctor told you that your HbA1c is too high and it needs to become lower, then your doctor was telling you that you have your glucose metabolic health was probably a little bit impaired. So 1.5 Ag is a longer term glucose metabolic health indicator, just like in humans would be HbA1c. So what we saw here, and this is directly related to the feed, is that the grain fat beef had a little bit higher amounts of these than the grass fat beef. Now, I want to preface this by saying is that this doesn't per se mean that the grain-fed animals were, per se unhealthy. It just means that as compared to the grain-fed animals, the grass-fed animals had better glucose metabolic health, which could be due to the fact that they have an increased ability to move, so they have more ability to roam, and probably because they weren't fed any grains. But there are certainly nuances, because what we see a whole spectrum even across feedlot systems. We did an interesting study, for instance, with bison in loose confinement in a feedlot, and they actually looked more similar to the grass-fed animals, or the animals who were finished on pasture. But here we had a comparison of feedlot finished beef with grass finished beef. And then in the TCA cycle metabolites, so these are the mitochondria, and the mitochondria are really the health, or the life of our cells. And again, this was quite stunning to see, is that I've done this work on endurance trained athletes, and we see this upregulation of these TCA cycle metabolites in endurance trained athletes. And we saw the same thing in the pastured cows. They look more athletic, not just like we find in humans, right? Humans are just another mammal too. So we find these interesting parallels between athletic endurance trained humans and pastured cattle on one hand, that show a similar metabolic phenotype. Now, homocysteine was higher also in the grain-fed beef, as you can see here, and lower in the grass-fed beef. Homocysteine in humans is associated with reduced cardiovascular health. So higher levels of homocysteine indicate reduced cardiovascular health. This has also been studied in other mammals, specifically in lab rats. But this can just be viewed as a further indication that as compared to the grain-fed animals, the grass-fed animals were probably metabolically healthier. Higher homocysteine levels in humans have been associated with increased risk of heart disease. Not sure yet how its presence in meat relates to human health, and if it all does, and how meaningful these differences are. But it is very interesting to see that, there is a difference there, which I think previously has remained unrecognized. Another are markers of oxidative stress. The animals out on pasture, they had higher amounts of uric acid, which is a major intracellular antioxidant. Also, 4-HNE indicates higher oxidative stress. So again, this was higher in the grain-fed animals, but it is important to note is that there was also variation amongst these. So I must admit that some animals experience maybe more stress than others, which could also be just an indicator of acute stress or prior to slaughter. But this does indicate that overall, the grass-fed animals experienced less oxidative stress, which is in line with other indicators of metabolic health. And final insight into animal metabolic health came from bile acids. So bile acids, they especially glycocolate has potential anti-inflammatory effects. And it is important in uptake of glucose and fat-soluble vitamins and fats from the rumen microbiome. And what was particularly interesting is that we were also able to detect these into the meat, and this was higher in the grass-fed animals, and it likely relates to better glucose metabolic health and better fatty acid profiles. So, because we know this bile acid is important in the uptake of those. So it's a further clue to metabolic health. And I presented about 10 metabolites so far of the animal health, and I could probably talk about this for 2 hours, but I'll spare you that because that's more interesting for a geek such as me. But there were multiple indicators showing that, hey, animal health in grass-fed animals was improved compared to the grain-fed animals. So does a healthier animal equal healthier meat? Let's find out. Nutrient density is a term that we often hear now. It basically talks about the concentration of nutrients given for instance, a 100 gram or a 4-ounce piece of meat. Phytochemicals means that it's plant-derived compounds. I told you earlier, there's half truth to the statement, you are what you eat. About 50% what circulates in our blood as humans is related to the foods that we eat. Well, for the cow, it is probably similar. Cows are what they eat to an extent. So when we start with phytochemicals, so plants, they respond biochemically to sunlight, moisture, nutrients, and they produce these phytochemicals. At moderate amounts, these can have anti-inflammatory effect, antibacterial effect, antioxidant effect. They can help with potentially management of chronic disease, though we must certainly study this in the context of animal foods. But what we have been finding out is that these phytochemicals are picked up from the forage and directly transferred into the meat and milk of animals. And we've been seeing some quite significant differences there, seeing that the animal is in fact what it eats. So we can be quite confident that these plant phenolic compounds are indeed from the forage, because that is their main source. If we look at here, hyporic acid is a main indicator of phenolic intake, so polyphenol intake. And polyphenols, we often think of this in the context of plant source foods, such as blueberries and carrots and things like that. But they actually appear in animal source foods too, especially when the animal is fed phytochemically rich foods. We found this to be twice as high in the grass-fed animals. Catechol sulfate, 4-ethynyl phenol sulfate are all downstream metabolites. These also become enriched while in our body or in the body of the animal. They can have potentially anti-inflammatory and antioxidant effects and improve both the health of the animal, and the next question is, do they improve the health of us? But we saw that these compounds were overall higher in the pastured animals. Now, the next slide illustrates that there is indeed a decrease in phytochemicals during concentrate finishing. You already see after 60 days, it's roughly cut in half because that is directly related to the phytochemical richness of feed. Now, we do have some interesting findings that we found in bison, and for today I focused on cattle, but actually feeding more phytochemically rich forage or feed in feedlot, such as alfalfa or other phytochemically rich byproducts, agri waste, can actually kind of rescue this. So that is important to note, is that it is certainly not black and white, and but there is a decrease in phytochemicals overall, typically during concentrate finishing, which is basically a decrease in antioxidants. So there is a direct connection between food and forage. This was actually nice work that was done in France in 2010, where they looked at the milk polyphenol content. So polyphenols in milk here on the left, forage polyphenol content here on the x-axis. Way up here was grassland pasture. Grassland pasture, we know, and this was from mountain pastures, they were very rich in polyphenols. And there's a direct relationship with the polyphenol content in milk, which higher amount of polyphenols in cows, if intake in the forage, equals higher polyphenols in milk. And it was much reduced when even that grassland pasture was dried and fed as hay, and the lowest amounts were found with the corn-based TMR, which is because that is fairly low in polyphenols. Again, indicating that with alfalfa feeding and higher amount of supplementing and this was work done in Italy, feeding alfalfa in a feedlot actually can get this number back up for at least for the carotenoids. So that is certainly very interesting, and one way to improve potentially the phytochemical richness in grain finished systems or total mixed ration systems. Vitamin metabolites then. again, we find differences here that directly relate to the forage. Alpha tocopherol is vitamin E precursor, best known for the antioxidant effects. We found that to be threefold higher. What was particularly interesting, and I had not come across yet as much in the literature, is that vitamin B3, niacin, which is the common form found in nature, was 9-fold higher in the grass-finished animals. Nicotinamide, which is also B3, but that is a supplemental or more synthetic form, often not occurring in nature, was twice as high in the grain-finished animals, which was likely due to the fact that, or is due to the fact that they received a supplement, a vitamin and mineral supplement, whereas the grass-finished animals only had a salt supplement. We also found that choline was 1.2-fold higher in grass-fed beef. It's an essential nutrient rich in forages. It's important for brain function, muscle function, and liver function. On the other hand, we found higher amounts of pantothenate, vitamin B5, another essential nutrient which is rich in corn. So some grain feeding can potentially increase that. So that is good to know. And we found the same thing in gamma tocopherol, really, another common supplemental form. So higher amounts of alpha tocopherol, the form that has a little bit more biological activity in humans. Gamma tocopherol can be converted in the body to alpha tocopherol, but it's a common supplemental form rather than the form that we typically utilize more readily in our body. Now, the effects on omega-3s are very well known. We found DHA, EPA, and ALA, alpha-linoleic acid, are omega-3 fatty acids. We found those to be 3 to 10-fold higher in grass-fed beef. And important to note is that unlike humans, herbivores readily convert linoleic acid, or ALA, into longer chain omega-3 fatty acids such as EPA and DHA. So humans have a slow conversion rate of ALA to EPA and DHA, but the animal does so more readily because ALA is rich in forages. EPA and DHA are important for brain function, heart function, and so these were higher in grass-fed beef. We also know from randomized controlled trials, oftentimes I hear this all the time, people say, well, it's very modest compared to a piece of salmon. Yes, true, but we still, when we do 8-week randomized controlled trials, we see EPA and DHA going up after consumption of pasture-raised animal foods. So they can contribute to overall dietary intake. And finally, I want to say very long-chain saturated fatty acids, similar to the long-chain polyunsaturated fats. Very long-chain saturated fatty acids became enriched in grass-fed beef. And at the time, we were also focused on omega-3s, and I hadn't really thought about this yet, but it obviously makes total sense that if you start enriching very long-chain omega-3 fatty acids, that the same thing might happen with saturated or monounsaturated fatty acids. Now, oftentimes we hear saturated fat is bad for you. Well, there's many saturated fatty acids, and the longer chain saturated fatty acids are actually associated with a decreased risk of cardiovascular disease. Now, those become enriched in when animals are fed high amounts of forage. So that was a very interesting finding that I was not aware of, and I arguably most consumers are not aware of either. So these were some of the major findings that we get from our metabolomics analysis, comparing beef from various systems. And while we found that improved phytochemical richness and enhanced ability to engage in activity, numerous indicators of animal health improved, and as well as the biochemical richness or the nutrient density of meat. But I do want to preface this with saying that our data does not indicate that grain-finished beef is unhealthy. There are many studies to suggest that consuming grain-fed beef as part of a healthy diet is perfectly compatible with good health. And there's also tremendous variation among systems. We see that because different feeding regimens in a feedlot result in different nutrient-dense pieces of meat. But what our data does indicate is that there's additional health-promoting compounds in animals when they finished on biodiverse pastures. And feeding more phytochemically rich feedstuff, as we're finding out in some studies now, like alfalfa, or enriched environments in feedlot systems can help reduce this gap. Like I said, we did some work with bison, and bison, they typically need a lot more room in a feedlot. and they consume less grain. Well, we actually found that the bison in the loose confinement, they were creeping up towards the pastured animals. So there's a whole spectrum. But to simplify it, typically what we see in our work so far, phytochemical richness of meat or the nutrient density of meat is improved when animals have access to biodiverse forage, when they're grazing a monoculture, especially in the case of like we have more overgrazed fields, the phytochemical richness is decreased, and when finished on concentrates, we see this further decrease in there. And I want to pull it all back, and this is my last slide, is that the bigger picture, what motivates a nutrition scientist like me to study this? And why do we want to do the systems research? Well, diets link environmental and human health. And I'm particularly interested in linking the fields of food production, nutrition, and population health. By improving the health of our soils and plants and animals, do we nourish ourselves, and do more agroecological production practices also benefit human health. And with that, I'd like to acknowledge everyone who has worked on this, various colleagues across different institutions, and also a great thanks to the farmers involved in this project. And by that, I would like to open it up to any discussions.
Thank you so much. I have learned a lot that I'm going to be processing for quite a while. So thank you. You mentioned that there has been some increase or improvement. There have been some studies done on additional feeding of animals in confinement situations. I certainly kind of aware of feeding seaweed, feeding other alfalfa, obviously you mentioned, but I'm just curious what kinds of things have been used in feeding trials, and what have those been showing?
So there has been some studies, and they've been done less in the US, but definitely more so in Europe and Asia. Is like the idea of feeding more. Here in the US, we feed a lot of grain, right? Grain byproducts. But in other countries, there has been also feeding of phytochemically rich fruits and vegetable waste in small amounts. And one project that we're starting, and I jokingly call it red wine milk, is that what if we feed limited amounts of grape byproducts from red wine industry, right? That often ends up in a landfill. What if those are fed in small amounts to the animal, obviously, in the amount they can handle? Do we see this increase in phytochemicals? And so there's definitely opportunities to also maybe include in small amounts, and by working with ruminant scientists to see if we can kind of, further increase the phytochemical richness there. So those are some of the things that we're looking on, but it can also be like, peels from different fruits and things like that. I know almonds are fed to dairy cattle in California, right? Almond hulls. So.
Well, that actually leads me to also ask, have you been doing any research in, I recognize we're talking about grain and grass fed, but have you been doing any research with different diets in the grain eating livestock, the monogastrics like chickens and pigs? I'm curious about that.
Yeah, we're just starting. We're just starting with we have a project going on right now with chickens. If we that were in confinement, but an enriched environment with additional phytochemically rich feed, actually more alfalfa. So it will be interesting what that shows, and we see some parallels with the beef. One thing though to highlight, and I think compared to monogastrics, we know cattle, they still spend a lot of time on pasture, right? Early on in their life. So they start off usually with higher amounts of phytochemicals, whereas my guess would be if we look at like sort of the monogastrics that maybe are born in confinement, right, versus those that are out on pasture. My guess would be the difference would be a little bit more dramatic, but that's just my hypothesis.
Well, it does. It actually, I am curious. I've had some conversations in Vermont about the number of folks who are doing a pastured pig. And I know that that's, all over our region as well, that folks are doing pastured pigs and also sometimes alternatively feeding them as well. And yeah, we've never quite gelled yet on projects around that, but it's certainly something that's of interest is what's the difference with those animals versus a straight confinement grain-fed pig.
Absolutely.
Yeah. So I did have a question from the group. Just a question about clarifying that dry hay has a decline in grass-fed benefits versus fresh pasture. Can you talk a little bit more about that?
Yeah. So what several studies have indicated is that when you harvest the grass, right, and then you store it, you dry it, that there's a loss of phytochemicals, about three to four-fold reduction in those, even if it comes from the same pasture. So, and yeah, that is just something to be aware of. I mean, we have seasonality in pasture-based systems. It is the way it is, right? So other studies have nicely illustrated too, and we're finding that too, is that there's this increase in phytochemicals. When the grazing season starts, it hit this peak in like the summer and into the fall, and then it decreases again. And I think that's why traditionally why in indigenous peoples, there usually was a lot of, or traditional agricultural societies, the harvesting season was done at the end, right? And that is also when the meat is most phytochemically rich. And yeah, we've been seeing that in some of our work too. Or there's some ways around it, yeah, potentially by feeding some of the like what we're seeing so far at alfalfa, but I'm sure it can be with other plants too, that you can kind of rescue this decrease a little bit in the winter. But it's something to be aware of, and we can try to but circumvent a little bit, but yeah, it often is the way it is.
Do you think that that's an argument for the hay in a day model that was brought up earlier in the session?
Could you clarify that? Because I think I may, in what context it was mentioned, because I think I may have missed that.
Oh, you may. Okay. So hay in a day is typically the cut it, bale it in one day approach to try to keep those nutrients, before there's too much wilting, before there's too much loss.
That makes sense. Yeah. I mean, my guess would be is that there is indeed then a reduced loss. Yeah. We haven't studied it yet. I'm not aware of anyone that studied the impact on meat or milk, but yeah, that would be a great way of potentially looking at that and see how that impacts it.
Related to this, I also have a question about baleage and curing, and would baleage actually retain those phytochemicals because it doesn't cure as long?
Yeah, so that's what studies suggest, that you still have a loss of phytochemicals at that point. Yeah, but like if you have silage, then you still see that loss. Yeah.
So just to kind of wrap it up, I'm sort of curious, like where is this going next? I recognize this is a body of work that is still very cutting edge and new. There's got to be lots of holes that you're looking at filling in terms of information. Where are you going with the next work?
So we recently started work with the Bionutrient Institute, which we dubbed the Beef Nutrient Density Project. So we've done this on a few farms now, a handful of farms. And we're seeing some relationships between the health of soils and the plants and the animals and the nutrient density. But in that project that we launched, we try to gather data from 250 farms across the US. See, can we make these connections? Do they hold up in a larger data set? And what are some of the principles that farmers use, both in feedlot systems, because feedlot beef is feedlot beef refining, and in pasture-based systems, I think both have their role. What are some of these principles that signify best practice? And it seems so far that some of these more, agroecological or sustainable practices also benefit the health of the animal and the nutrient density. And while that perhaps is not surprising, there's a big lack of data on that. And without data, we cannot really make A, good statements, and B, impact food policy, and C, the farmer may be well aware of that, but also when it comes to small to mid-sized producers, and it's really also what we're focusing on, data, marketing that to consumers, is very important, and having sort of an evidence base to start off with, like, hey, what are the best practices? Can we come up with these principles? So anyone interested in learning more about that project, feel free to shoot me an email or just Google Beef Nutrient Density Project and my name or Bionutrient Institute. And yeah, you can find more information about this.
Excellent. Thank you so much. Thank you so much for sharing your time with us today. This is amazing data, and we really appreciate you joining us for the consortium today. So thank you so much. Thank you. And I just want to encourage folks to go away and come right back at 4:15. We will be doing our research priorities reevaluation for the coming year, and we would love your impact on that, your input on that. So thank you so much. Thank you, Stephan.
Thank you so much. All right.